or kind of the other way around?
The ones that I recruit, I want to be mathematicians. I think they can pick up the biology on the fly, but I need people who can prove results, and if they can't do that, then they're lost. I mean, it is mathematics first and foremost. So and I think it's a bit easier to pick up the assumptions of the biological systems. I mean, it's hard to get in the detail. You kind of need, for some problems, you need a biologist to lean on. But actually, the stuff I'm talking about today, it's quite possible to get right into it without much biology. But I've found the ones, like one of my students who's maybe joining us, he's in Germany now as a postdoc. I mean, he did a master's in group theory at Sydney Uni before coming to me, and he did a great job.
Yeah. So it's in algebra and algebraic topology and representation.
I think people, well, I'm happier if they know some group theory, but as long as they, I don't know, I've never taken anyone who's like an analyst or something. Maybe I should, just to learn some, but I mean,
it's pretty easy to shy away from PDE.
I think, like, the thing is, I want to be able to give problems for them to solve that you could give advice. Help with, you know.
Because you mentioned University of Virginia, and we have a colleague, I think Ilir, at our place at UFRJ, who he studies lots of other things, but profinite groups, and I think he did a postdoc in Virginia.
Okay.
Yeah, but he just studies that. So I think, but it's if he was looking for a problem, I could suggest something in this area to him. But
it's a good idea.
I'm
getting emails from people saying they're waiting to be let in, Stefanel,
Yes. I'm letting everyone in. At 1 second.
Yeah, that's right.
Well, just
Don't worry about it. They'll be fine. They'll survive. So how did you end up, Andrew, in Brazil?
I was doing, well, I had a Brazilian girlfriend ultimately, but was but I had some contact. I'd visited Brazil previously, and for summer school workshop thing. And I was doing a postdoc in Europe, and the opportunity came up. And so I was on a postdoc, not with Paulo, but where Paulo is, and then then there was a job opening here in Rio, and so I put my name
That's
in.
great. Yeah.
But and so at that time, Brazil was really taking off. It was there was lots of investment in science and innovation and technology, and it was very, there were, you know, really interesting perspectives on it, which have tightened up a little bit since then.
What happened to your beard?
My beard. Or his beard.
Yes.
Hi, Marika.
Hey.
Like my hair was out here until yesterday, so yeah, I've been taking advantage of the quarantine to let it all go.
Yeah, I did the same. My beard was a bit shorter before quarantine, but
yeah.
Okay, so I think 1 second. Well, so I think we can start record also, and Paulo, welcome to everyone. And first I introduce Paulo Piccione, that is going to tell us words from the Brazilian Mathematical Society. Please, Paulo.
Okay, good morning, everybody. It was a real pleasure to be invited and to open this talk. Let me first briefly say a couple of words that, as probably all of you know, we're going through a difficult time in Brazil and in science, especially in science. But at the same time, this situation has opened a range of possibilities of meeting people, especially on the internet. And I have to say that since all this has started, I have witnessed a very impressive growth of organization of events, and many foreign researchers have managed to enter a little bit in our scientific life, as will happen today. Today, our guest is a professor from the Western Sydney University, which is okay in Australia, of course, and he's Andrew Francis, and I am very pleased to have him. And he's probably can be considered a new friend of our scientific community in Brazil, I also understand that this is part of a graduate course that Stefanella and some collaborators are organizing at the Federal University of Rio de Janeiro. And the Brazilian Mathematical Society sees all this with very proudly that this is happening. So thank you to Stefanella for organizing this, and your collaborators. I don't know exactly who they are, probably Andrew Clark also. And thank you to our guest from Sydney, Andrew Francis. And I wish all of you a very fruitful talk, as it seems to be the case, actually. So thanks.
Thank you, Paulo. And now we have our local Australian, Andrew, please.
So thank you very much, Stefanella, for the opportunity to contribute. I'd like to, and thank you to Paulo and to Andrew. I'd like to present Andrew Francis from the University of Western Sydney, or perhaps Western Sydney University. I'm not sure if the nomenclature has changed. Who will, and to give us the opportunity to, I guess, increase these collaborations between Brazilian and Australian mathematics, which, and there are some links that I've already seen, but to this time, not enough. So I'm very glad to be able to contribute I'm very glad that we're all contributing to this collective collaboration. So thank you to Andrew for coming and speaking to us today. I'm not sure.
Yeah. Okay. So I think Andrew, Andrew can start his
I can
talk.
start my share. Well, just before I start sharing, I'll just thank you for those really kind words. Thank you for the welcome, Paolo and Andrew, and thank you, Stefanella, for inviting me. It's, I mean, I would love to be in Brazil with you, and so I hope one day that's possible. In the meantime, I was saying to my colleagues that, you know, although we're all in isolation in Australia, like I think many places are, people are being individual, working at home, but somehow that equalizes our relationships, because you are no more distant than some of my colleagues are who would normally be down the corridor. So we have these opportunities to get together. So thank you very much for the invitation. Stefanella, should I go ahead and share the slides and start?
Yes, please. You read. Yeah, thank you. So yes.
Okay, so can you see the title slide?
Yes. Yes.
All right, so this is a talk which is going to cover a broad survey of mathematical ideas and in phylogenetic networks. which is a fairly recent area of development, I would say, in the last couple of decades, maybe even only 1.5 decades. So it's not going to delve too deep into particular problems, but give a flavor of a range of different problems in mathematical networks, phylogenetic networks. So before I start, I want to give you a little bit of background about how I began in this area. It is now about 6 years ago, I guess I shouldn't say 5, but I originally was an algebraist, and perhaps arguably still am. I began in algebra. I did my PhD in algebra in finite reflection groups. I studied Coxeter groups and UFRJ algebras. And this diagram is as it happens, a rooted phylogenetic network, which describes the evolution of my research life. So I was an algebraist, and then I began working in epidemiology. So I looked at the spread of tuberculosis and studied drug resistance evolution in tuberculosis. That was really heavily with a biologist collaborator of mine. Then we together started working on genome organization. So that's this evolutionary branch point here. I hope you can see my mouse on the screen. I was looking at the way bacterial genomes are organized. So where the genes are positioned on the bacterial genome. The bacterial genome is a circular chromosome, single chromosome, and evolutionary processes lead to genes being clustered. And so we were modeling that. And while we were modeling that, and I was learning more about the biology, I saw that there were ways to look at this through the eyes of group theory. So I could see that you could model the evolutionary processes on the genomes with a finite reflection group, with a symmetric group, or even a type B Coxeter group, the hyperoctahedral groups. So this is this hybridization here between algebra and genome organization here. And so I worked on this for some period, and I still do. I have a PhD student who's still working on that, and I've just had a paper published this year on that. So this then led me to think about phylogenetics. And the reason it led me to think about phylogenetics is that some of the motivating questions in genome organization are phylogenetic questions. They're questions about how related different species are, or can you establish a distance between two bacterial genomes? And if you can you build a tree or a network that explains their history? So I think of that as algebraic biology down here. Algebraic statistics sort of crept into it. I won't say anything about that, but that's been quite interesting too. And phylogenetics is an offshoot, and phylogenetic networks is now probably what I do most of the time. Well, most of the time I do administration, to be quite honest, but most of my research time is on phylogenetics. So the question when you think about networks is, why not trees? I mean, phylogenetic trees have been around since at least the early 1800s. Charles Darwin, in his notebooks, drew phylogenetic trees to explain the way he thought species were related. So they are a beautiful combinatorial or mathematical object, and there are lots of very difficult and interesting questions, but they're just a little bit too simple to represent some parts of what we know about evolution. So people started to wonder, like in particular, about certain processes in evolution which are not tree-like. So I'll just read out this quote from Ford Doolittle. It's already a 20-year-old quote, and a beautiful picture here of different bacteria, eukarya and archaea. He said that phylogeneticists will have failed to find the true tree, not because their methods are inadequate or they've chosen the wrong genes, but because the history of life can't be represented properly as a tree. So people were looking for the true tree or the right tree to relate species. And to do that, they would sequence a gene or maybe a set of genes to try to construct the true tree. And what he's saying is that they will have failed to do that, not because they didn't have good enough methods or they'd chosen the wrong genes, but simply because life is not tree-like. So there's an alternative view, which is that even if it's not tree-like fundamentally, it could be more or less a tree. It could be something like this. This is a quote from Eugene Koonin, a central tendency tree with transfers between the branches. So these transfers, let me give you some examples because it helps to see this. So here's a picture of a tree from a 15-year-old paper. These points on the tips are leaves. They're the species that we're looking at. And back here is the common ancestor of all of these species down this yellow dot at the bottom. And you can see bacteria and archaea separating out in this tree. These branches that go sideways between the branches of the tree represent events like hybridization. So when two species merge to form a new species. So that's not something you can represent on a tree. And they also represent horizontal gene transfer, or lateral gene transfer, which is quite a common phenomenon in bacteria. So the genes or any DNA genetic material from one species can be inserted into the DNA of a separate species. It's very difficult to imagine that happening in our genomes, but this happens in bacteria quite regularly. And it's a very important mechanism because it's thought that drug resistance, for instance, is passed around in that way. So this emerging awareness that networks needed to be studied was concretized, was stated in Cohen's essay. This is a beautiful paper I encourage you to look at. I don't know if you can read the link down the bottom of the reference. It's a beautiful title of a paper, I might say. Mathematics is biology's next microscope, only better, and biology is mathematics' next physics, only better. So he had a number of challenges for the use of mathematics in biology. And one of them was to replace a tree of life with a network or a tapestry that will represent lateral transfers, like that's horizontal gene transfer that he's referring to. So here's a grab from a website which is dedicated to networks. It's called Who's Who in Phylogenetic Networks. It's run by a group in France, and they track the papers appearing each year. So you can see that there was a big jump, 15 to 17 years ago, and it's sort of motored along since then. You wouldn't exactly say it's exponential, but it's a very active area. I've always thought this underestimates, because this is only the network papers. Of course, phylogenetics themselves is a much larger area. So what is the goal? The goal is to understand and represent evolution that is not tree-like, which is to say most evolution, or maybe a significant portion of evolution. Maybe most is too strong a word. So in particular, we want to represent non-tree-like evolution when you cannot place distances on a tree. So you may have, for instance, a matrix of distances between your organisms. This is a common thing to do. You give me two species, and I'm going to construct a metric on the set of species, and I want a distance between them. And then I want to build a tree that represents those distances. It's actually quite common to, it's almost always the case that your distances will not fit on a tree. So a network might be used to represent those distances. You might want to summarize information when a set of gene trees are incongruent, which means that they don't match properly. So one way that people construct species trees is to construct a set of gene trees, and then they try to use a consensus function to get a species tree, like a true tree, or they might draw a network which captures all of those gene trees in them. Or finally, when you might want to physically represent true events or actual things that happened in the history of these species through, hybridization or horizontal gene transfer. HGT stands for horizontal gene transfer. So there are at least 3 good reasons to look at networks. Here are some algorithmic problems. So that are in a book by Dan Gusfield, again, like from 6 years ago. So I'm not really, I do algorithms, but I'm more a straight mathematician. But there's a huge group of computational mathematicians or even computer scientists that try to understand complexity of algorithms and build better algorithms for working with these problems. So Dan Gusfield is more from an algorithmics angle, and his list of problems include how you can reconstruct plausible histories, including tree-like and non-tree-like events, how you can determine the minimum such events needed to derive the sequences. So your data, if you like, are a set of species, and you want a network to represent the relationships, but you want to do it in an efficient way. So there are infinitely many networks. So I haven't defined a network for you yet, but I will shortly. But there are infinitely many, and so you want to choose or find one, or find a way to find one which is somehow parsimonious. You might want to enumerate a range of possible histories. So maybe there's a bunch of equivalent or equally plausible histories, or you might want to characterize the properties of plausible histories. And I would add a couple more. I'm interested in defining classes of networks that capture some important property, whether it's a biological property or hopefully also a mathematical property, and then to study those classes of networks to see if they can tell us something about the biology or give us access to these algorithmic questions. So I'm going to focus on a broad family called explicit networks. That is, networks whose goal is to represent an actual history rather than summarize a metric on the space. So you can think of I mentioned before, if you have a set of genomes with distances, you might use a network to represent those distances. I'm going to focus on networks that describe an evolutionary history in them. So the events, the vertices on the network are going to represent real events. Well, supposedly, that's the goal. So I'm going to talk about some classes of networks that capture key properties and some underlying problems, some key ones like identifying trees in networks, identifiability, I'll explain, and reconstruction. And there's 3 books that I think are all pretty good and a good place to start reading. Okay, so a definition. So for me in this talk, I'm going to mostly talk about binary rooted phylogenetic networks. So this is an example of one. It has several different types of vertex. First, it has a root. That's the one at the top here. It's got in degree 0 and out degree 1 or 2. I allow it to have out degree 1. And the other vertices are either in degree 1 and out degree 2, like these. These are ones that would be at home on a tree. So they're called the tree vertices. Other ones like this one, which are in degree 2 and out degree 1, they're reticulation vertices. They could represent either hybridization or horizontal gene transfer. And finally, the leaves, in degree 1 and out degree 0. So the orientation of this graph is down the page. So it's a directed graph. It's a directed acyclic graph. And I won't be drawing arrows on the edges, but they're implicit, I hope. I will also mention a little bit unrooted networks. So this is a binary unrooted network. I should say binary refers to the interior vertices, which are all degree 3 in a binary network. Many of the results I'm going to talk about work for non-binary networks, but for simplicity, we'll just talk about this. So here is a beautiful beautiful binary unrooted network. There's no root, but otherwise it's got leaves and interior vertices. When you don't have a root, you can't distinguish between tree vertices and reticulation vertices. You don't distinguish between in degree and out degree. This one is a non-directed graph. Okay, so before I launch more into explicit networks, I will just say something about data display networks. These are networks that aim to represent distances among the taxa. So you may have a set of species, maybe they're genomes, or maybe they're whatever they are, and you've got some metric on your set of species. Your metric could be derived from DNA, or it could be the number of morphological traits that are shared or not shared by your species. It doesn't matter. You've got a matrix of distances. You want to know if these distances can be represented on a tree, and if so, which tree. So there's this beautiful theorem by Peter Buneman, which originally came, I believe, out of the linguistics literature. So Buneman's theorem is the 4-point condition, and it says that you can place a metric on a tree if and only if this condition is satisfied. So the condition is that the sums of these two distances, sums of these pairs of distances are equal, except one is smaller. So let me just give you an intuitive explanation for what this theorem is saying. Here is your tree down here. This is an unrooted tree. The root doesn't play a role. It doesn't matter where the root is here. If you look at the distances that are represented by these sums, if we just take this first sum here, duv, that's the distance from u to v on this tree. That's a path around this left-hand edge. And then I'm adding it to dwy, which is this path around here, right? So think of that one. This one is the small one of these three, because it doesn't include the middle edge. The other ones, u to w, the second distance here, this one, is distance u to w plus the distance v to y, right? So it's going to be bigger than the other one. And then finally, the third one, distance u to y plus v to w, is going to be the same as the second one, but bigger than the first one. So this is it's a 4-point condition because it tells you exactly when your metric can be put on a tree, and it reduces to sets of size 4. You just have to look at all the subsets of size 4, if you like, to check if your metric can be placed on a tree. And of course, it can be placed on a unique tree as well. So if the 4-point condition fails, then there are algorithms, neighbor joining is the most famous one, that will help you give a tree that's, the best available, I guess, for some definition of best. So there are algorithms that take your metric and produce a tree. And this is a hugely commonly used approach. If you don't have a tree metric, but you still want to get a tree, you use neighbor joining to do it. But you could also use an idea called neighbor net, which gives you more information. So here's an example of a neighbor net. So this is a result of David Bryant and Vincent Moulton from 2004. It's a very heavily used bit of software. And here's an example of, just if you search for neighbor net or search for citations of David and Vincent's paper, you'll find lots of these examples. So this is telling you about the relationship between various families of goats. And you can see clusters of closely related goats and more distantly related goats in this picture. And I guess what you're getting in these parallel lines, these boxes in the middle there, is you're getting the uncertainty. And perhaps there was hybridization in there, perhaps there was some probably not gene transfer with goats, but certainly hybridization is very possible, that messes up the signal, messes up the metric signal. So you get bits like out on the bottom there, bottom left, which are more tree-like, there's almost nothing going on, and other parts which are definitely network-like, where there's probably a lot more mixture of the species. So explicit networks use reticulation vertices, and I'm going to have them all directed. And I'm going to talk about three key themes. The first is reconstruction. It's the really big problem, I think. If you've got some data, can you reconstruct a network uniquely? I mean, what conditions do you need to reconstruct a network uniquely from the data? What data do you need, and what conditions do you need? Displayed trees are trees that appear in a network if you remove some other vertices and edges. And I'll talk a bit more about them in a minute. Identifiability. This is a related problem. If you have genetic data which was generated by a model on a network, so you've got some evolutionary model, probabilistic model, which generates genetic data, can you ever recover the network that produced the data? So it's slightly different from reconstruction, because in reconstruction you're just trying to find a network uniquely. With identifiability, you're assuming it's arisen under a network. This is a more philosophical problem, really. Is it philosophically possible, like perhaps with a long enough sequence? Maybe the sequence has to be impossibly long to do it, but is there a theoretical possibility of you being able to reconstruct your network if your data arose from that network? And lastly, this is the more combinatorial part of it, really. Can you find features of the networks that help you to answer these questions? I think I'll probably spend more time on the last one. I am, see, I've said it myself. All right, so there really is a zoo of phylogenetic networks about. And so here's a picture, two pictures of different classes of networks from Hewson, Rupp and Skornavaka's book. Please don't try to read the names of all these networks. It's not really important. It's just important to see that they're there. And I think it's amusing that this book uses, I mean, this is a phylogeny, right? There's a network of networks to try to explain the relationships. And I mean, what this tells you is that networks and graphs are very natural concepts, and they represent something more fundamental about the way we see the relationships between things. Then Dan Gusfield's book has this picture and several others, also with different families of networks. Some of them are in the Hewson, Rupp and Skornavaka book as well, and some of them aren't. He was focusing on the ancestral recombination graphs, which are in here. And lastly, there's a picture by Mike Steele, already 4 years ago. This is his representation of the way the networks interact, the way they're related and nested within each other. So I'm going to mention most of the networks in this picture and talk a little bit more deeply about some of them. So we want to establish a language and try to find which properties of a network help us capture something fundamental. Because basically, as I said, there are too many phylogenetic networks. There are infinitely many for a given set of taxa. So we have to focus our attention a little bit if we've got a chance to make some inference. So I'm going to talk at least about the following three classes: the level of a network, tree child networks, and tree-based networks. And then I'll end with some identifiability and reconstruction talk. So just to remind you again, what is a network for me here? This is a directed acyclic graph with a root, with leaves, and with tree vertices and reticulation vertices. Okay. So what's the level of a network? Level is an early concept in the study of networks, which tries to capture how close a network is to being a tree. So trees are level 0 networks. This one is a level 1 network because the only thing that stops it from being a tree are these 2 cycles here that you see. Of course, they're not cycles because this is a directed graph. But if you forgot about the direction, that would be cycles. So it's called level 1 because when you look at the blocks in it, the blocks are the maximal biconnected components. So a biconnected component is a component of the graph, a maximal part of the graph, for which every pair of vertices is connected by 2 paths. So biconnected, connected in 2 ways. So these little blocks here are biconnected components, and they're maximal because you can't add any other vertices to them and remain biconnected. So these biconnected components can be removed by deleting just one edge from each of them. So that's the level 1 part of it. So the level is the level of the maximum level of all the blocks in your network. So in this case, it's level 1 because each of the blocks can be eliminated by deleting just one edge. Another way of thinking about it is that each one has one reticulation. Maybe I'll go back. Each one of these blocks has just a single reticulation vertex at the bottom. So those are equivalent characterizations of level. So here is an earlier example. So let's figure out the level of this network. That is the maximal biconnected component. You can't, if you took a smaller part, then you would be able to add some extra vertices and still be a biconnected component. So that's it. And it's level 4 because there are 4 reticulation vertices in there. I think there's this one here, and then there's 1, 2, 3 here. And you can play around and delete 4 edges and get yourself a tree. Of course, I have to warn you, it may not be a rooted tree. You may have to forget direction, but you can sort of treeify the network with 4 deletions. Okay, so I'm going to talk about a couple of problems with networks. So the first is the tree containment problem. How do you tell if a particular tree is displayed by a particular network? So what does display mean? A tree is displayed if it can be obtained from the network by deleting arcs. So arcs are directed edges and redundant vertices, so degree 2 vertices. So here's a very little simple network, this one here on 3 leaves. And you can see here that if you dot out these edges here, then you've got the tree AB against C. So this is notation, this AB/C, just to indicate that AB is this pair and C is separated from it. But you can also get these are called triplets. You can also get the only other 2 triplets displayed in this network by choosing correctly the edges. So for instance, if you, this is the same network, I've just drawn this sort of curved, but if you delete that one, which would be this one over here, and delete that one, then you've got AC together. So A and C are now together here against B. So you can get all 3. This network displays all 3 possible trees. So the question of whether a tree is displayed by a particular network is actually NP complete in general. But oh, who did that? That's good. Did you highlight that, Stefan? Oh, somebody put a highlight on. That's very clever.
No, I didn't.
Oh, wow. Very good. So somebody's able to do that. That's pretty cool. Pretty dangerous. You could just get in there and anyway, that's fine. Okay, so tree child networks are, so I'm going to talk about some specific classes now. So if you put that blue line there, and if you know how to get rid of it, please do that so that it doesn't. I don't know how to get rid of that. Anyway, it won't matter. Tree child networks, they've been around for 11 years, defined by Carano, Rossello, and Valiente. So here's a definition of a tree child network. Oh, thank you. A rooted network is tree child if every internal vertex has at least one tree child, one vertex that is a tree vertex as a child. So for instance, this vertex here in this network, or this segment of a network, has a tree child here. This one is a reticulation vertex, but it has at least one. That's all it needs. Here's a reticulation vertex. It has a child that's a tree vertex, so that's fine. On the other hand, here, this reticulation vertex does not have a tree child vertex. It has only a reticulation child. And this tree vertex here has only reticulation children. So it's not a tree child network. So tree child networks are really beautiful. What I love about this area is, if I go back to this property, this is a really simple property for a network to ask from a network. Just this property that every vertex internally has a tree child. And yet it gives you some powerful consequences. So this theorem says that every vertex has a path to a leaf that only consists of tree vertices. Okay, so you've got a network. It's got a lot of tree vertices, reticulation vertices. It's clear when you think about it, of course, because every vertex has a child that's a tree vertex. You follow that path. It's got a path to a leaf. It's not a difficult theorem, but it's kind of important biologically, because it means that if you're interested in evolutionary signal, so you've got some process which starts at the top end, and you get a duplication which goes down 2 branches, and there might be separate mutation. And then at each vertex, you've got things happening and going on down. When you have a tree vertex, what happens to the genes, the genetics, is that it's duplicated and travels down the 2 descendants. And so there will be mutations along those paths, but they start the same. Right? So you're keeping a signal from the parent in the 2 children. When you have a reticulation, you've got genetics from 2 parents coming in and producing a third. You don't know what is retained in the third, in the child of that hybridization or that horizontal gene transfer. You don't know what's there. Some of it might be there from both parents. By definition, we would say something non-zero is kept, but you're going to lose a lot. So when you have a path from a vertex to a leaf consisting only of tree vertices, you're saying that the signal from each vertex should be present in the leaves, you hope. There's a sort of a better chance, at least. It also forces some constraints on the networks themselves. Really strict mathematical constraints. So the number of reticulations is at most 1 less than the number of leaves. So X is the set of leaves, and the number of reticulations is strictly less than the number of leaves. And here's the proof, is like another one-liner. If you take the tree vertex paths, they do not intersect. So if you take them from the reticulations, there's non-intersecting paths from reticulation vertices to leaves. So the number of reticulations is strictly less than the number of leaves. Okay, so this is another reason I love tree child networks. Magnus Bordovich and Charles Semple proved just a short time ago. This was not a major result in their paper, but this lemma, you know how sometimes it's really a lemma. You know how sometimes in mathematics, beautiful little lemmas are what people get remembered for, or drive a lot of innovations, because it's seeing something clearly. So this is a reduction result, because it says that in a tree child network, you always have a cherry, which is just a pair of leaves that are descended from a single parent, or a reticulated cherry, which is a pair of leaves like this. They they have parents who are related like this. So one of them's a reticulation vertex and one of them's a tree vertex, and they're connected like that. So that's a reticulated cherry. They made up that language, of course. So that's great. Every tree child network has such a thing. But what's better is that when you reduce a cherry, when you delete one of these and replace it with a single leaf, or you delete that edge there between those two, U and V, you still get a tree child network. And why that's important is because it just allows you to use induction. You can now use induction on your networks, because you can say, give me a tree child network. Let's suppose my property that I want to prove is true for this many vertices. And then now, you know, I take a tree child network with one more, and then I delete it, and I can use that inductive argument. And I've certainly used that in my own work. So I'm going to just throw out a few more tree child results because you know, this is a survey talk, but you know, I don't expect you to understand or remember any of this. But you remember I said earlier that in general, the tree containment problem is NP complete. In general, is a tree contained in a network NP complete? But it's polynomial for binary tree child networks. So that's nice, computationally advantageous. This one is beautiful. If you have a binary tree child network, so Stephen Wilson has been working on networks. He's from Iowa, and he does written a lot of beautiful results in networks over a long time. If your tree child network has no redundant arcs, then you've got 2 to the R displayed tree. So R is the number of reticulation vertices, reticulation vertices. If you think about it, if you've got a reticulation, you can, and you want to eliminate it, you want to display a tree, you can choose one side or the other. So there's 2 choices for each reticulation. So the most possible trees that you could display is 2 to the R. That's the upper bound. You cannot get more than 2 to the R. So this result is saying that for binary tree child networks where there's no redundant arc, you get the maximum. You get all the possible trees, which is a great result. So a redundant arc is just one. An arc is redundant if there's an arc there and there's some other path between the 2 vertices. So it's an intuitive definition of redundant. And networks like that are called normal. So Wilson called them normal. And then he had another result, which is related to the reconstruction problem. I think that if he defined another class called regular networks, which are normal networks but compressed. So I didn't want to put in all the technical definitions because you're not going to remember these things in 5 minutes time. But compressed means if you have a tree vertex, so a tree like that, so I don't know how I can sort of do this with my hands. You've got a tree and an edge going up there to a reticulation vertex. So you've got a reticulation down to a tree. That middle arc is kind of redundant, it's not in the technical sense, but he compressed it by just shrinking it down. So then he has networks that are not strictly speaking networks in my sense, because they have vertices with in degree 2 and out degree 2 are possible. So that's a compressed normal network. So those networks can be recovered by their displayed trees if they're normal networks which are compressed. Sorry, I hope that's not too mind-numbing and hand-wavy. Okay, so there are a few other classes. I'll just throw them out here. This one, tree sibling networks, are those for which every reticulation vertex has a tree sibling. And, you know, these were played around with, like people come up with these families of networks in the hope of capturing something important, right? And then they try to prove results, they prove results. And, you know, we're still in that process of exploration, I think, in this field. So this was interesting, that the tree containment problem, while polynomial for tree child, was was NP for tree sibling networks. So, you know, somehow like you lose a little bit of control when you just drop to tree sibling networks. Tree sibling networks contain the tree child. So another one, more classy, reticulation visible networks. So I am going to talk about visible vertices a bit later, so I will come back to visibility. A vertex is visible if, so you take a vertex, if there's some leaf for which every path to that leaf from the root goes through your vertex. So imagine an evolutionary process that gives me a leaf that might go through this vertex, but might also wander around it and go down to the leaf through some other way. Then this vertex might be invisible in the data down below. It might not have been seen. If the evolutionary process going down here, if all the information here can be tracked a different way, then this vertex is pointless, if you like, it's invisible. So a visible vertex is one if every path from the root down to at least one leaf has to go through that vertex. So then you're going to get the signal in the leaf. So reticulation visible vertex networks are those for which every reticulation vertex is visible like that. And it means that the reticulations might have some substance to them. They might carry some signal in the leaves. And tree child networks are special in that sense. They're the networks for which every vertex is visible, not only the reticulation ones, but all of them. And you've got a nice tree containment result there for reticulation visible networks. So that's kind of like a bit of landscape. I'm gonna delve into this family a little bit because it's one that Mike and I worked on and still do to some extent. So a tree-based network is, I think, perhaps a slightly more intuitive definition, at least I hope it is. A network is tree-based if you can obtain it from a tree just by putting arcs between the tree arcs. So you start with a tree and you add additional arcs between those tree arcs. You're not allowed to put them between the reticulation arcs that you've just added in. So this example, so I'll give you another characterization right now to make it easy. It's tree-based if and only if it has a spanning tree. Okay, a spanning tree, sorry, not complete. A spanning tree whose leaves are the leaves of your network. Of course, any graph has a spanning tree. You were going to say that. You probably all shouted at me with your microphones off. But a tree-based network is one that has a spanning tree whose leaves are the leaves of the network. Okay, so if you have a tree-based network, then it will display, if it's based on T, so imagine this tree here, this solid line tree here, this network is based on this tree because these arcs here are going between the tree arcs. It's also based on this one because these arcs are going between tree arcs. It's also based on this one because these arcs are going between tree arcs. Okay, so in this case, this tree is based on all of those networks. Oh, sorry, this network is based on all of those trees, and in particular, it displays them all. But it's not true in the converse. So this network here can display both AB against C and A against BC. So you have to be a bit cunning to do this. These are the same network, these two. I've just drawn them slightly differently, curves differently. So you can see here that B and C are together. So this is this triplet here with A off further away. And then there's these other arcs over here. But those arcs mean that it's not based on this tree. It's not based on BC because these arcs, these reticulation arcs, are not between the edges of the tree. So this one displays both, but it's only based on one of them. So tree-based networks include all of those other families that I've mentioned: tree child, tree sibling, reticulation visible. So it's a large family. And that's you can see this now. I think actually I've mentioned all of these classes. So now you know what this diagram means. Tree-based is the class on the outside, and we saw reticulation visible briefly. You may have already forgotten that, but there's tree sibling, tree child are in there, and the level ones and the normal networks are in there as well, are all tree child. Okay, so you might ask, are all networks tree-based? And the answer, of course, is no, because that wouldn't be a very interesting class if they were. In fact, Mike and I got started thinking about this problem when we saw this blog post from Leo van Jersel, who's at Delft. So he has a nice blog, and so I encourage you to have a look at this. It's not only his, there's a group of people who curate this blog, the genealogical world of phylogenetic networks. So this example is not tree-based, and if you stare at it a little bit, you will see there's no spanning tree whose leaves are just A and B, but it covers all of the vertices. It's a spanning tree. It's not possible. So it's not tree-based. But there are smaller examples. This is perhaps the minimal example of a non-tree-based network. A network with one leaf is not a very interesting network, but mathematically it's, of course, perfectly acceptable. All right, so here's a little quiz for you. Two networks. I don't know if I can conduct a poll in Zoom, but I'll just have a look at these two and think about whether one of them or both of them or neither of them are tree-based. If you just stare at this one, so most people, I won't waste your time with this, but most people, when they look at the left one, think that's not tree-based because the tree is these outside edges, and these two edges are joining reticulation arcs. They're joining the additional arcs, but it is in fact tree-based. I'll tell you why in a minute. This one is not tree-based, and basically, I guess, if you think about spanning trees, you can see that you really are stuck. But the problem is really this little position here. You see these three vertices that I'm trying to circle. There are three reticulation vertices. You can't have a spanning tree that includes them all. So there's actually, I'll get to some results which make that quite explicit. So, sorry, this one is tree-based because you can draw the tree going down here to U, down here to X, and down here through V and A. So you draw the tree going down through X and then V, and then this edge here is just connecting two tree edges, and this one as well. So it's not always completely obvious if something is tree-based or not. So some necessary and sufficient conditions. So if you're tree-based, then each reticulation vertex, like this one, must have at least one parent of out degree 2. So this is what rules out that example on the right on the previous slide. You can't have that. And if you have every reticulation vertex with both parents being tree vertices, then you're guaranteed to be tree-based. But neither of those conditions are both necessary and sufficient. Needed a little bit more information. So our result was the following, that the network is tree-based if and only if there's an admissible subset of arcs. So an admissible subset just comes from thinking carefully about what happens at these vertices, really. So if you think about a tree vertex, if you're going to have a spanning tree, you must have this edge up here, this arc above that tree vertex in your spanning tree. Okay, so that's a T for true. So you must include that. And down below, if one of those is not in the spanning tree, the other one must be. Okay, So because if they're both not, then you've got a leaf here, which is not a leaf of your original network. So if one of them is not in your base tree, then the other one must be there. And similar logic, but the dual logic, if you like, is with the reticulation vertex. If you've got a reticulation vertex, you must have this out arc here, because otherwise this will be left hanging as a leaf in your, which is not a leaf of the network. And if one of these is true, if one of these is in your spanning tree, the other one cannot be, because they can't both be in a tree. So they're very simple sort of logical rules, and that's a computationally tractable problem, because you can relate that to an instance of two-sat, two-satisfiability, which is a logic problem. So here's an example of this algorithm. It's already been completed, but you can go through these vertices and say, okay, here's a tree vertex, so that must be a T there. Here's another tree vertex, that must be a T there. Here's a reticulation, that must be a tree there. And once you get down to this vertex, you've got to have that true, and that means this must be false here. Likewise, what have we got here? This vertex here, you've got a true there, and that one must be false. And that's a problem for you, those two falses, because you've got a contradiction. You've got an incoming edge here that must be in it, and you've got nothing coming out. So it cannot be tree-based. So there are a number of different characterizations. Most of them are graph theoretical characterizations. So if you're a graph theorist, you might love this stuff. We characterized it with the family with admissible subsets. Lucian Zhang from National University of Singapore shortly after found a nice way to characterize it graph theoretically. He said they're tree-based if and only if there are no reticulations with both parents also reticulations, and no pair with a zigzag path, what he described as a zigzag path. So if you like, it's like a bunch of reticulations stacked up in a zigzag with the tops here on each side. These ones, the R0 and the RK+1 reticulations connected by the zigzag path. So it's tree-based if and only if there are none of these substructures. That's a nice graph theoretical way of characterizing them. And here's a bunch more to do with bipartite graphs. So Lucian also constructed a characterization with a graph with tree vertices and reticulation vertices on two sides of the bipartite graph. Laura Jetton and Leo van Yersel produced a graph of omnions and reticulations. Omnions, I think, I haven't asked them, but I'm guessing this came from the Terry Pratchett books, the Discworld books, some of you may know. Omnians were a particular religious group, but the definition here is a reticulation that has a child that's a reticulation or something like that. Is that what it was? I've forgotten the definition, sorry. Anyway, they use a bipartite graph with some very specific vertices, and together with Charles Semple, Mike and I did another one with all the vertices on each side, and we constructed a characterization using that bipartite graph. So, and there's one more, which is, it's a tree-based network. If you can partition the vertex set into a set of vertex disjoint paths, each ending at a leaf. So obviously you can partition a network into a set of vertex disjoint paths, any network. It's tree-based if you can do it such that each path ends in a leaf. And I guess I'm going to just I think I was going to prove something about that in a minute. But basically you can see why this would be true, because if you have a set of paths which end at leaves that partition the set of vertices, you can join those paths together to form a spanning tree. That's basically the connection there. As long as you've got your paths ending at leaves, you've got a spanning tree. So for tree-based networks, you have, because tree sibling networks are inside the tree-based networks, tree containment is also NP hard for tree-based networks. It's also NP hard to decide if a network is based on a given tree, unfortunately. However, I was quite excited to see Momoko Hayamizu's paper last year, where she tried to rank tree bases. I think this is a really nice question to think about. Like, you've got a network, and if it's tree based, your biological intuition is that that tree is kind of like a fundamental species structure, and you've got reticulations moving between the basic structural species branches of the tree. So, but if it's based on many trees, how do you pick one? And so Momoko and her collaborator tried to use probabilities on arcs to rank tree bases using a likelihood method. So she's a graph theorist and used some probability theory to look at that. And I thought that was a great question. Another thing I might say about this slide, the paper by Anaya et al., there's a long list of authors down there on the bottom of the slide. They published this result in one of the top mathematical biology journals. This is a group of undergraduates doing research led by Megan Owen, so you can see Megan's name there, Owen, and Catherine St. John, so you can see St. John name there. So Megan and Catherine, they're at the City University of New York, and they have this nice program of working with research with undergraduates. And so they took some of these problems and they got a group of people working together. So I think they were all undergraduates when they started this project, and I don't know how long it took them, but they produced that result very quickly, because our paper only appeared in 2015 and theirs in 2016. So, I mean, I really liked that they were able to, the problems are kind of accessible. They're certainly not easy, but some of they're easy to state, perhaps. And I think they used some nice computational tools. So they had some good programmers perhaps in the group. Okay, so one thing that I was interested in was, so not all networks are tree-based, but, you give me a network, it may not be tree-based, but you can make it tree-based quite easily by adding leaves to it. So you can think about tree-basedness as a sort of, you can think about that as being a core, and other networks all being related to tree-based networks. And maybe you can measure how far they are from being tree-based. It might give you some information about the processes or data that you're missing. So this is a project I did again with Charles Semple and Mike Steele. And you can think of any number of fun distances. One of them is the number of leaves that you need to add to make something tree-based. One is if you have a spanning tree, the minimum number of additional leaves you have, which are not part of the original leaf set, or if this number of vertex disjoint paths that partition the vertices of the network. So if it's tree-based, you will have one path for every leaf. If it's not tree-based, you'll have some additional ones. So this last index, P of n, is counting the additional paths that you need to cover the vertices. And the theorem is that they're all the same, which is nice. And I've got a quick sketch of the proof. I'm sure you're all suffering a lot, so I don't want to go through it in too much detail, but it's really a nice little combinatorial argument. If I mentioned this part of it before already, if you have n disjoint paths or p disjoint paths, you can join them to make a spanning tree with the right number of additional leaves. And if you've got a spanning tree, you can add leaves to so if you've got a spanning tree where some of the leaves are not the ones at the bottom of your network, you can add extra leaves to those other ones to make it tree-based. So that's the T. And finally, if you've added some leaves to make it tree-based, you can decompose it, and you get this circle of inequalities to prove they're all equal. So I want to mention just briefly an unrooted story, unrooted tree-based networks. This is one nice property of tree-based networks, I think, because many of the other things, definitions or classes of network, you can't move to the unrooted world. But you can with tree-based networks, because you can just say it's tree-based if there's a spanning tree whose leaves are the leaves of your network. So this was joint work with Katharina Huber and Vincent Moulton from the University of East Anglia a few years ago. So we showed that, in fact, all level 4 networks are tree-based that are proper. So, I mean, maybe I won't bother about that definition, but basically all sensible level 4 networks are tree-based, but not all level 5 ones are, which is, it's kind of surprising that so many of them are tree-based. And this result came out shortly after, which is a beautiful result that tells you there are only 2 non-tree-based level 5 binary networks. That's them. And 2 of these authors, Marika Fischer and Katharina Vicker, are here in this session. I saw them creep in. They're from Greifswald, the University of Greifswald in Germany. So thanks for coming, Marika and Christina. Yeah, so that's a lovely result. There's a little bit of computational. So Vincent and Katharina are both sort of mathematical computing people. So I'm not somebody who's very good at thinking about NP completeness or anything. So I defer to their authority on this. But we were able to show that the decision problem for the unrooted case was NP complete. Now, if you remember the rooted case, it was polynomial. We used the 2-sat to show it was polynomial. So, and it's quite easy to see that it's NP complete. Even I could understand this argument. If you have a cubic graph, any cubic graph, so that is a graph all of whose vertices are degree 3. And if you add 2 leaves to just one edge, so you've got an edge here and you add 2 leaves to it, then you've got a phylogenetic network. And if it was tree-based, if you could decide if it was tree-based, and then you found a spanning tree, that would solve the Hamiltonian cycle problem, which is NP complete. So this problem is also NP complete. So the interesting thing is that the rooted case is polynomial. And so this is joint work with Marika Fischer, who, as I mentioned, is already here, joining us from Europe. You can relate the unrooted and the rooted problems quite nicely. So the unrooted network is tree-based if and only if it can be rooted on the midpoint of an edge with orientation specified to give a tree-based rooted phylogenetic network. And so you might say, well, this doesn't explain why one of the problems is polynomial and the other one is NP complete. And the difference is that when you go from the unrooted world to the rooted world, you have to specify orientations on a lot of edges. And there are 2 to the r edges that need that. So that's an exponential number of decisions you need to make. So you would think that the unrooted problem might be simpler or faster, but at least I would, not being a computer scientist. Computer scientists in the audience would probably say, this is obvious, go away. But, you know, we all have to learn from each other. Ah, a quiz. All right. So I didn't tell you about this, Stefanella, but I had prepared a quiz. And now I have to find it. If you can scan a QR code, do that. And now I'm going to see if I can get to the chat. Can I get to the chat?
You should be able to see the chat.
Yeah, maybe I'll have to stop sharing the screen. Where would I find the chat?
On the bottom.
Yeah, I'm not seeing that. I'm just going to. Oh no, it's over here.
Yeah.
Okay, I'm going to paste a link. So if you've got a QR scanner, you can do that. Otherwise, you can follow that long link in the chat box. And I've got a little quiz for you. It won't take long. Please give it a try.
Okay. Tree chart.
I have to see if I can find it. The last one is a hard one, the number of visible vertices. But now, can I see the outcomes? I just learned how to do these forms yesterday. My wife teaches English to migrants, and she said, "Oh, you should use Microsoft Forms." I've never thought of doing this, but it was really quite easy. So I can see there are 3 responses. Everyone's got the first question right. 3 of you have got the second one right, and only one of us has got the third one right, and that's me, because I. Yeah, the last one is hard. Okay. Was that interesting? Not many people of you have participated, so I guess it's, I know it's late at night for everyone. So
not for us, it's late for you.
Yeah. It's nearly midnight here now. Anyway, let's get back to the talk. Never mind. So you can play with that later and at your leisure. All right, so I wanted to return to normality, normal networks.
Can I ask you a question? Can I ask you a question?
Please, please.
Yeah, when you say the biological intuition, if we have some species that the DNA can mutate by insertion, like you say at the beginning, you work on that. That could rule out the tree structure if we look at that.
Yeah, because that's right. Because the insertion is insertion of DNA from another species.
Yeah.
So then you've got the other species, of course, was evolving on its own track.
Yeah, yeah.
And so you've inserted some DNA from that other evolutionary history into your one. So it's no longer tree-like. Tree-like would be just mutating what you have down through history. Insertion means you're bringing some new DNA in.
So just, no, I'm thinking about it because we done last week some biological lab. And so what virology use some app they're available to them in the net, on the network, you know, like, and this app carries some assumption, you know. The assumption is that there is always a tree is rooted or not rooted, but they don't take, it seems to me, I mean, I'm still new to that, but it doesn't seem to me that they are taking truly in account this hypothesis of insertion. But maybe, I mean, you know, I'm just new to that, so maybe it seems kind of important to take.
So I think, without wanting to, you know, go too far into it, but I think there is a lot of work. People publish trees. Biologists publish trees because they are simple. They probably explain most of the variation, and so people often will ignore that, or they will look at a tree for, like, if there was an insertion of one gene, and you were to sequence that one gene, you would get a different tree. The tree would follow a different path. So that's what they talk about when they talk about, you know, contrasting gene trees and then trying to get the species tree. So the paradigm that people are often in is that there is a species tree that you can draw, and those other things are just, you know, small details which are difficult to understand, difficult to know about. So I think it's true that I think there's a lot to do in making good inference, and I'm quite excited about this work with normal networks, because I actually think that we're onto something with normal networks now. If I just segue back onto this, Stefanella, and we can talk more about that some other time.
Yeah, okay.
Yeah. So you probably, you know, I mentioned this a little bit earlier, but a normal network is a tree child network without redundant arcs, so without arcs where there's another path between the two edges. And they have the property that every vertex is visible. If you remember, tree child networks have every vertex visible. They are precisely the networks that have every vertex visible. That means that they are capturing good, they've got a good chance of capturing the signal from the vertices. All right. So we've just proved earlier this year that every phylogenetic network has a canonical normal network. So there is a map from the set of all networks that, like there's a projection onto the set of normal networks, and it's a beautiful, simple projection, which means that you can talk about the normalization of a network. So it's not a difficult thing to do. Here it is, the algorithm very simply. You just take the visible vertices of your network, then you construct a cover digraph of those vertices. And a cover digraph is a directed graph. It requires you to have an order on the vertices, which is going to be given for us by the structure of this network. And then you suppress the vertices of degree 2 and you're done. So here's a network. I'm going to color in the visible vertices first, right? So the first visible vertices are the leaves and the root. Remember, I'm looking for vertices for which there's a leaf, for which every path between that leaf and the root has to go between the vertices, through that vertex. So these ones, these 4 I've just colored in now, are just immediately above the leaves. So if I think about this vertex here, this root, this leaf, any path going up to the root has to go through this one, right? And the same with this one here. Every path from this leaf to the root has to go through that one. So that's a visible vertex. Same for this and this. But that's not all, because as you can see here, this one is also going to be visible, because when I come up here from this leaf, I have to go through that one. So these 3 are also visible. If I'm here, if I've come from one of these leaves, I have to go through this vertex. If I've come up this side, I have to go through that one. But this one is not visible, because if I've come from one of these, I could go around that way. If I've come from one of these, I could go around this way. I don't have to go through any of these ones that are not filled in. So those colored ones are the visible vertices. So step 1 is I take all those vertices. Step 2 is I construct the cover digraph using the network. So I've drawn an edge between the vertices. If there is a path between them in the network, that's sort of minimal, that there's no other vertex sort of halfway along that path. And so I've got a few degree 2 vertices and I just ignore those. And this is a normal network obtained from my non-normal network. That original network here is not a normal network. So that's the normalization process. So I'm excited about this because I think of lots of really interesting mathematical questions. I want to know about the fiber of a normal network. If you're used to thinking in algebraic topology terms, you might think of space fibering down a bundle down onto a smaller space. So this is a loose analogy of that language, but you've got, what's the pre-image of this normal network? What networks are in that pre-image? What relationship do they have amongst each other? I don't know. I was able to prove a few little things, but I don't know, and I think that's a good question. So what this little result says is that normal networks are these ones in the middle here. They're quite a small class, but they're kind of, I think they're a canonical class. I know I've talked a lot about tree-based networks because it's something that I like and so on, but I now think normal networks are really a bit special, and I think we can focus on those when we get deeper into problems. So this is an exercise that I'm not going to stop to let you do, but it's quite easy to if you're bored, draw a network and construct its normalization. Yep. And one more advertisement. This is being recorded, so you don't have to write down this link. But if you search for Philo Sketch, Dan and Mike have produced a lovely bit of software that allows you to play with networks. It's not published yet, but it's available to look at and use. So I've got not very much more time, but I just want to touch on identifiability and reconstruction briefly. So this is going to get a bit probabilistic. So I apologize if that's not your flavor. It took me quite a long time to understand this concept of identifiability. So the problem is basically a reverse engineering problem. When you think about an evolutionary process on a tree or a network, you've got a tree, you've got the graph theoretical structure, and you've got a model of evolution, which you may not know quite what I mean by a model of evolution. I'll explain what I mean in a minute. And when you're in the real world, you don't have the network or the tree, you have the data. You only have the data, sequence data. Let's just restrict our attention to genetic sequences. So your scientific question, your scientific question is, what's my tree or network? Your mathematical question is it theoretically possible to reconstruct the network or the tree? If you know that, if the data evolved on a network, can you find it back again? Of course, it might have evolved on some other process that's not even network-like, so you're assuming something about the network. So in practice, what happens is we're going to fix the model of evolution and ask whether we can identify the network, assuming the data arose under the model. So here is an evolutionary model. This is the sort of thing I mean by the evolutionary model. You have a set of character values. So a sigma could be anything, characteristics of something could be, but for me, it's going to be the nucleotides, the DNA nucleotides, A, C, G, T. And down here, I'll just draw your attention to this array. This labeled by X, Y, Z, and W. The rows are labeled by the species. So you can see in the tree here, I've got X, Y, Z, W, they're the species. If you read along each row, that's a sequence, the genetic sequence of that species. The sites, when I say for each site, I'm talking about this column. So what I'm starting with is a genome alignment. These genomes are aligned, and I know that that's one site that is related on this tree or network. Okay, so each site has evolved independently. So what's happening is for each site, I choose a value at the root. So it's going to be like an A, C, G, or T. And then then it goes down the edges, and I change the value with some probability mu, and to each different alternative. So the probability of changing from A to C is mu, and so on. And I go down here, and I keep going, and as I get to each tree, it splits, and then there's still a mutational change, and I get a sequence alignment. I get a character value at X, a character value at Y, a character value at T, and at W. So I get the values at this site when I've started at one. And then I do it again for another site, and then I do it again, and I get this aligned set of sequences. Now, trees are great. If your data evolved under a tree, then you can identify the tree, at least in theory. And Allman and Rhodes, Elizabeth Allman and John Rhodes from Alaska, have done a huge amount of work. And actually, if you're an algebraist in this audience, you'll be interested because they are quite algebraic. I think John was originally a topologist and Elizabeth an algebraist. And they've done quite a bit of work on algebraic evolution models. So look them up. So you can extend that model to networks. And how do you do that? Well, for the tree vertices, it's the same. You evolve down here and then you split, the same character goes down both sides and you evolve down each branch. The issue is, what do you do when you get to a reticulation? So when you get to a reticulation, one model is you throw a coin with probability P1, you pick it to come from one side, and with 1 minus P1, you pick it to come from the other side. That's a very simple way to do it. And you can choose, you can have different reticulations at different probabilities at each reticulation if you want. So then you would construct your alignment site by site. So you do one site, ACGT or AGT, whatever, and you keep going. And that's a common model to be used in identifiability for networks. And it's difficult to prove identifiability. And so Elizabeth Gross and Colby Long, a couple of years ago, I saw their paper and it got me interested in talking with Vincent Moulton about this. So they were able to prove identifiability for networks, but if they had quite a strong restriction on the networks, they had to have only a tree, but with just one cycle, only and it had to be a bigger cycle than 4 vertices. So, you know, it has a network that is quite simple, they're able to do identifiability for. So Vincent and I looked at an alternative model of evolution, which we actually think is more realistic. So you have your reticulation vertex has a probability P, like before. But what you do instead of evolving at each site individually, you choose a displayed tree in the network at random. So you start with a tree. And then for each site, you evolve on that tree. And so you keep going site after site on one tree. But after each site, you throw the dice and decide whether you're going to change tree. So you change the whole tree at once. And then if you change the tree, you evolve more sites under the new tree until you keep throwing the dice or spinning the coin until you change tree again. So the difference is that you're generating a sequence of sites under one tree and then a sequence of sites under another tree. And the reason I think this is more realistic is that if you have had horizontal gene transfer, you're getting a chunk of DNA coming. That DNA has all evolved under one tree. So you're going to get stuff happening, your own species with its DNA evolved under a tree, and then you're going to get a chunk of DNA that evolved under a different tree. And so it feels more natural than this site-by-site evolution. So typically you get whole genes that are moved around rather than like individual nucleotides. So we got, this is not completely our own idea, we adapted it from one by Balchandra That, who published it a little bit earlier, for pedigrees. So quite a different context. I'm not going to tell you what pedigrees are, but we could adapt it to networks. And so here is sort of thing that happens. You've got a network, and these 4 trees on the right are your 4 displayed trees. You've got 2 reticulations in the network, so there are 4 trees. And of course, the top 2 here are kind of the same tree, except they've got degree 2 vertices that are popped around here, and I'm leaving them in deliberately. So if you take different choices, so these are the ones, the edges that I'm deleting to get here. So what's happening in this alignment is I've got this site generated under tree 1, this under tree 1, and a bunch under tree 1 all together. And then I change, to T2. And then I continue like that, and then I change to T3. So my alignment looks different because it's aligned under consistent blocks for a single tree. So this is an identifiability theorem, which is kind of wordy, but let me read the theorem first. So if you have two networks with omega reticulation vertices, there's a condition. So this Sn and Sn prime, these are the embedded spanning trees. These are the ones up here with the degree 2 vertices that I've left there. So if every tree of those is an X tree, so it has leaves from the leaf set, and if the number of those trees for each of the two networks is 2 to the omega, that's the maximum possible, and we know that happens with tree-child networks, then there is a length L and a probability P, so this is the length of the alignment, such that the distributions of the alignments obtained under the network being equal implies that these sets of trees are actually equal. So you may say, well, that's not identifiability, and it isn't really yet, but it's the key kind of technical result. The distributions of alignments being equal implies the sets of trees are equal. And that's kind of the key thing, because we have this other result which says the tree-child networks are isomorphic if and only if those sets of trees are equal. So this theorem tells you that this characterizes tree-child networks being isomorphic, and then that's going to link back to the identifiability result. So it's also true, as I said, for tree-child networks that you've got 2 to the omega such trees. So the top part of this slide is the same statement, but basically the theorem is the bottom one. The class of tree-child networks, there's a little technical glitch. The root doesn't form an arc with the reticulation vertex. That's identifiable. So tree-child networks as a whole, large family, are identifiable under that model. So that to me is a nice result, which points to tree-child networks being a very important class. You won't get a result like this for tree-based networks. There's too many of them. But tree-child networks give you a chance. And this is also why I think normal networks are a thing to look out for, because normal networks are just a special class of tree-child networks. Finally, I know it's late, but I'll just say a few words about reconstruction results, because there are some nice thoughts here that Mike Steele and Charles Semple have worked on with Peter Erdos from Budapest. So again, we know that you can reconstruct regular networks because they're determined by the display tree. So if you have the display trees, so this is a kind of reconstruction result, this one due to Wilson. If you have all of the display trees, then you can get the regular network back. Erdos, Semple, and Steele gave 3 nice it's a nice little paper. I think you should read it. It's referenced there at the top, published just last year. 3 philosophical approaches to reconstruction. So the question is, how can I reconstruct a network? The first one, Fabio Pardi and Celine Skornovaca proved that, well, they took the approach of not of only trying to reconstruct up to isomorphism or some equivalence class. So they just said, we're not going to get the exact unique network, but we're saying that the network is within this class. So that's one philosophical approach. The second one, number 2, is limit your attention to only a certain subclass. And you say, well, if the network that this data evolved on was say, a tree child network, then I can identify it. Then I can reconstruct it. But it could have been something else, so I don't know. But if it was just this class, then I'm okay. And what Peter and Charles and Mike did was they allowed additional information beyond what you would normally use. And so that's also a really cute approach that I hadn't seen before. So they defined another class of networks. See why I said it was a zoo at the beginning, really is. Orchard networks, they're the ones that can be reduced by cherry reduction to a single vertex. So you remember I mentioned what cherry reduction was. You can reduce the cherries or the reticulated cherries, those ones where there's a sort of a branch like between like that. So tree child networks have that property, but orchard networks is a bit of a bigger class. And they defined extra information, meaning the ancestral profile. So on each vertex of the network, they want the number of paths to each of the leaves. So they want quite a bit of extra information. It's cheating really, but they were able to prove that up to isomorphism, you can get your orchard network back. Okay, so that's all the content. I'm going to just sum up now. I haven't said much about algorithms at all, and there's a huge amount of work by computer scientists and other computational mathematicians that, I mean, some of the people I've mentioned have been heavily involved with that, but there's a whole world out there. My view of the mathematical goals is to provide tools and provide ways of understanding networks that are hopefully going to be usable eventually. I think the field is still a way away from that goal in the context of explicit networks where reticulations and so on are actually given. I think we're still a long way away. But that's kind of the reconstruction and identifiability question. But before we can answer those, we need to do the maths to be able to describe the families of network. So that's what I think the mathematical goals are. So I want to finish with something to wake you up, which is that, you know, trees have applications absolutely everywhere. And I mentioned Peter Buneman's 4-point theorem right at the beginning, which came out of linguistics. And I'm actually doing some work with a linguist right now using phylogenetic trees. But I think there may be other applications of networks. So I mentioned at the beginning that I started work in UFRJ algebras, and I don't expect you to know what they are, but they're quantum deformations or Q deformations of group algebras of Coxeter groups. There you go. But they're closely related to finite reflection groups. So this picture, if you can see the date on it, it was scribbled in 1996. I drew this in my PhD time. This is the algorithm I was working on to construct elements of the center of the UFRJ algebra. The columns are conjugacy classes of the Coxeter group, and the rows are the lengths of elements, and these components are little equivalence classes that I defined, and I realized you had an algorithm to construct elements of the center by working up these edges. So I drew this. It's not been published, it's just in a drawer. but look at this. This is like, the picture is almost the same, it's just upside down, of course. But this is the picture I showed you as an example from a paper by Buneman, with a sort of a central tendency tree with additional arcs. And when I look at this, I think, wow, you know, one day I've got to go back to the Hecker algebras, and with the results that that I know about now in networks, maybe we can, you know, finish some of those things I never finished when I was a PhD student all those years ago. That's speculation. So I want to end by thanking my collaborators in networks. There are people not here that I collaborate with on different areas, and there are people who I'm still working with who are on networks that are not here. But these are people I've finish some work with. I really appreciate all their efforts, and I appreciate some funding that I've been able to receive to keep my work going. And Stefanella, thank you again for having me, and thank you all for listening. And I'm sorry I haven't left you much time for questions, but I'll leave it there. Thank you.
Thank you very much. I think I will bug you with in the future with some more reference, because it seems very important what you point out. Of course, I have to do my homework in learning more math, but it was very clear about some of the fundamental questions, and this how is useful this classification of network, and how it's possibly linked to biology, evolution. It seems very crucial, because as I say, before all these algorithms that are ready to use, they come with some paradigms, and the universe is so much more, bigger than this paradigm. So this, it seems very crucial. I really like your model where you can jump from a tree to the other. So I'm looking forward to see a paper, because I really like it. And
okay.
So let's see if I think people get scared to ask questions, but if someone have question, you're welcome to ask question. We're here to, let's see if someone raised their hand. Yeah, people are
it's a long time to listen and absorb things. So
yeah, but so how do you come out? So I'll ask, how do you come out this idea to of jumping or switching tree? I mean, it seems very important when you have this insertion from another DNA, but how this? How
do you mean the
at the reticulated vertex?
Oh.
Instead to just the probability just to jump to another tree,
Oh, you mean the identifiability?
Yes, yes.
We have to give credit to Balchandra That for that. So that was his model on pedigrees that we sat down and I'll tell you how it happened. I was visiting Vincent Moulton in Norfolk, Norwich in East Anglia. And I said, look, there's this paper. I had a couple of weeks with him, and I said, there's this paper by Elizabeth Gross and Colby Long, and I'm really interested in whether we can do something on that. And he said, well, funny you should say that, because there's this other paper by Balchandra That, which I've always wanted to understand, and it relates to it. So maybe we can work on them both together. So we tried hard to understand Balchandra's paper and translate it into networks. And it was extremely difficult, I might say, but he had the idea to do that. And I think it was only after we had worked through and realized it was going to work that we started thinking that it made biological sense. So this is not the way you're supposed to do mathematical biology, right? You're supposed to ask what the biological model should be, and then try to work out the maths of it. In this case, we worked out the maths to get a result with a model, any model, and then after the fact we thought, well, actually it is actually an intelligent model. I mean, it's biologically reasonable.
Yeah, no, I think it's very reasonable. I wonder, because I mean, as you know, here we have many different viruses crossing each other, so, and we have, even with dengue, you have that the virus go through different kind of species. So it's an hypothesis that has to be taken into account, because it seems very interesting. I think we'll have to discuss more with that.
So I'll introduce you, Stefanella, to Marika, who's sitting here and with the toddler on her lap. She's also
hi. Hi, hello.
Germany.
Hi.
Hi. Yes, we also have this at some time toddler And, Renata last week had her child clean her mom.
Yeah, 2 hours is quite long with a 10-month-old baby.
Yeah.
But
We
luckily,
know
it's the middle of the day for me. It's not too late. It's 4 p.m. now.
Yeah. It's hard to accumulate a time schedule that is good for everyone. We try. So this is also why we record it, because there are some people in California that also for them was too early, and so it's hard. But
I have Luan here, my colleague from Sydney. Luan is here as well, I can see, and so you can see.
Okay, so you're not the only one suffering.
No. Oh, and there's a few others actually, Roger and Chad.
Everyone is welcome, and I really hope that we could have more exchange, because we really need some good insights like this here. Like there is a lot to do. We have a lot of different data, as Renata,
Yes. Just one question. I don't know if I'm talking. Yes.
Yes.
Okay. So as Stefanella told, I mean, I don't know if you ever saw this network building for viruses, because I mean, tree is very common for us, but this is the first time that I'm listening about this networking applying for biology, and that's I don't know if you ever heard about it or saw.
I haven't with viruses, and I guess so. I haven't worked on viruses, so I'm probably going to say something really stupid, but I have talked to my biologist friends about them to try to understand. And so my understanding is that they evolve really quite rapidly. So like, for instance, with COVID, they do produce trees to try to identify where outbreaks have come from, but locally. And they'll I mean, actually, it's such a short time since the outbreak started that it sort of makes sense. I don't know, you may know more if there is insertion in viruses. I just don't know, or hybridization.
Yes,
I don't know how they reproduce. I mean, I know they depend on the host DNA for reproduction and the host cell machinery, but that's about all I know.
Yes, I'm curious because in some virus we have the genomes as segments, and when you have the genome in this kind, it's quite often they mix the genomes, and it's like it's most like a hybridization also. I mean, and maybe in this kind of virus, this network will be more feasible and better than the tree, maybe. But I think we have to try.
Yeah, I think this why. Yeah, because
the challenge is the reconstruction. The challenge is getting enough information to actually, be confident that your network is representing something real. I mean, you can make a method to produce a network that doesn't mean anything. So that's the kind of the key scientific question that I think we're waiting for the right answers to. So the neighbor net that I mentioned earlier on, with the lots of branches going everywhere and big bunches of things, that's quite commonly used. And you're not saying that those vertices, internal vertices, represent anything biological. The network is a way to represent your data without suggesting that there are particular reticulations there. So that might be worth looking at, because that's kind of well studied, and people use that for inference. They use that to draw conclusions about whether there has been some hybridization, for instance. but they can't pinpoint where it was or what it was. And that would be nice. I think that's the kind of dream. But I think we're still a way away from that.
That would be an intermediary step. So before to do that, and there is like a black box.
Yeah.
And then to aim to a sort of network, you know, if I understand, no? This will be intermediary, no? So
yeah,
okay. Can you eventually, not now, but later, can you send us some reference that we can look example, you know, thinking about that
You're thinking about
we
the
are
algebra
learning.
problems.
Yeah, no, we're learning. No, yeah, we can ask local algebra, but you know, easiest the better to start with.
You want an easy maths paper? Is that what you're saying, Stefanella?
At the beginning, then we can go. You know.
All of my papers are easy. It's by definition. Look, yeah, no, I'll definitely send you some anyway. Yeah, I hope they're not too hard.
Okay, so other question, I think Mareke, you can speak up. You can speak up.
No, I just put a remark in the chat that because before you asked about links between Brazil and Australia or so, and I just wanted to mention that Balchandra Tate, who has been mentioned now in Andrew's talk multiple times, he is actually in Brazil. He has been there for at least, I don't know, 10 years or so. I met him in New Zealand. I collaborated with him quite closely when I was still a PhD student. And yeah, at some stage he accepted a professorship in Brazil. So he is in Brazil. I posted the link to his website. Oh, and someone here seems to know him.
Yeah. He's at UFRJ, apparently. Anyway.
Is that UFRJ? Yes, is our university? No.
Somebody in the chat has said that he's at UFMG.
Ah, UFRJ. Yes.
He's a colleague there.
Thanks so much.
A good fellow, but I have never collaborated with him, but a very good fellow.
This is Armando, is from UFMG. It's a university of Federal of Minas Gerais. This is Armando Neves.
Yes, that's the one. That's where he is, yes.
That's great. It's a small world after all. Andrew Clark there. And, yeah.
Okay. So thank you for the information. So maybe Armando, you will do your homework and connect.
No, I'm going to tell Balchandra he was cited.
Yeah, no.
Maybe he will see the.
Say thanks from me. It was good that he did that. He did that was a very difficult paper that he wrote, and so full credit to getting that done. It was a great piece of work.
Okay, so if there are no further questions, I'm going to release Andrew. Then it's over midnight. No,
that's right. Go to sleep.
But thank you so much. I'm looking forward to more. I think I'll bug you with more reference and basic questions, because I think there is a lot that very interesting things that we have to catch up over here. You know.
Sure. Yep.
Looking forward also to having you here and to come back to the network over there. It was very interesting in 2015. So.
Well, I hope we can do that one day in the not too distant future.
Yeah.
But thank you very much for having me. It was really nice to do that, and it's an honor to give a talk to you all. And
yes, and hopefully there will be more occasion. And to everyone, that I will put the recorded to our webpage, so so everyone can also access this later. Thank you very much once more, Andrew, and well, good night to you.
Okay.
And
good night. Yes.
In Europe.
And thank you very much, Andrew. It's very nice to meet you.
Oh, thank
nice
you.
to meet you all too. Yep. Bye.
Thank you very much to everyone. Bye bye.
Bye bye now.
Bye.
Bye bye.